Vaccines are killed, attenuated or antigenic preparations of microorganisms which generate immune response when introduced inside the body. The response develops in the form of antibodies and sensitized T cells. They provide immunity to the host against infectious organisms. Vaccines are used as preventive as well as curative measures. History of vaccinology. The concept of vaccination can be traced back to several years where there have been instances of preventive methods. In 429 BC, Thucydides, a Greek historian, noticed that patients who successfully recovered from smallpox did not catch the infection again. In 900 AD, Chinese discovered the process of variolation. In this process, the infected tissue from smallpox patient was inserted inside the skin or in the nose of a healthy individual. This prevented them from the disease. The process kept on spreading around the globe slowly and became worldwide by 17th century. With this background, most important contribution in the field of vaccination was by Edward Jenner. He developed the process of vaccination against smallpox for the first time. He noticed that milkmaids did not suffer from smallpox. They contracted cowpox due to the exposure to cows' infected udders. It caused mild illness, but at the same time provided immunity towards smallpox. Edward Jenner inserted the fluid from cowpox blister into the skin of an 18-year-old boy, James Phipps. Later, he introduced smallpox infected tissue, but the boy did not suffer from smallpox. Rather, he was immunized against smallpox. Eventually, the vaccine was developed. Later in 1880, Pasteur was working with chicken cholera following the principles of Robert Koch for germ theory of diseases. He discovered that old cultures of pathogen could not develop disease in the host. He tried to develop infection again with fresh cultures, but failed. He concluded that the older ineffective cultures must have primed the host to develop immunity, which prevented them from the subsequent infections. He developed vaccines against pathogens causing chicken cholera, anthrax and rabies. Edward Jenner is known for introducing the concept of vaccination, and Louis Pasteur is known for the development of first vaccine. These vaccines consisted of either killed or attenuated cultures, but there was another category of diseases which were caused by toxins produced by pathogens such as tetanus and diphtheria. Emil von Behring and Kitasato in 1890 explained that the serum from the individuals who have recovered from diphtheria could transfer immunity to healthy individuals. The serum contained antitoxin, the antibodies against diphtheria toxin. This opened the way for passive immunization. Alexander Glenay in 1923 developed a method for active immunization against diseases caused by toxins. He prepared inactivated tetanus toxin, now known as toxoid, by treating it with formaldehyde. Later, vaccines for diphtheria toxin was also developed in 1926. Application of all the above vaccines drastically reduced the mortality rate. Vaccine has saved largest number of lives in these years among all the pharmaceutical drugs. How a vaccine prevents us from diseases? Pathogens cause disease inside our body by invading our tissues or organs. The immune system of our body recognizes the antigenic structures on a pathogen and mounts adaptive immune response against it. This response is in the form of antibodies in humoral immunity and sensitized T cells in cell mediated immunity. Our immune system possesses immunological memory, because of which once the immune response is developed against any agent, it protects us from subsequent infections. This property of immune system is applied to provide immunization through vaccines. Vaccines contain preparations of microorganisms which are either attenuated, that is weakened, or killed. These preparations cannot cause infections, but can provoke immune response because the foreign structures which are present on these microbes are recognized by the cells of our immune system. The pathogenicity is destroyed, but immunogenicity is retained in the microbial cultures used as vaccines. The body mounts immune response against the vaccine preparation and develops antibodies or sensitized T cells. After vaccination, if the host contacts the same live pathogen which tries to cause disease, it will be destroyed by the pre-existing immunity developed because of the vaccine. Vaccination is done for immunization of an individual. The immunization can be active immunization or passive immunization. Active immunization. If the vaccine contains antigenic preparation, the host's body will elicit immune response against it. This is active immunization. Active immunization deals with provoking immune response against the introduced agent and developing an immunological memory. Any subsequent exposure with the antigen elicits heightened immune response and successfully eliminates the pathogen. State of active immunization is achieved naturally by combating a disease, where the immune system recognizes the foreign antigens and develops sensitized T and B cells. This is natural active immunity. Active immunization can also be done artificially by vaccines. Vaccinating individuals for various diseases has tremendously reduced the mortality rate. Active immunization for infants has served as a boon for mankind by reducing the infant mortality enormously. Healthcare providers have designed an elaborate schedule for preventing the infants from dreadful diseases. Vaccination schedule. Vaccination for children starts at birth. The vaccination schedule includes the list of vaccines which protect the infants from common infections such as poliomyelitis, tuberculosis, hepatitis, diphtheria, pertussis, tetanus, measles, mumps, rubella, Haemophilus influenzae, chickenpox and pneumococcal infections. Vaccination process has made numerous infectious diseases rare and eradicated some of them. Smallpox has been eradicated from the world and polio is on the verge of eradication. Pulse polio. Pulse polio is a program developed by Government of India to eradicate polio from the country. This was started in 1988. This program is carried out in the entire country at the same time. All the children in the country between the age group of 0 to 5 years are vaccinated against the polio by oral polio vaccine. This has effectively reduced the incidence of polio in the country. Herd immunity. Vaccination helps in developing herd immunity and protects the unvaccinated individuals also. It is also known as community immunity, where a large percentage of population is immunized against infectious disease and provides indirect protection to the rest of the community. When a majority of the individuals are immunized, the disease does not spread in the community. If the infectious agent infects an unvaccinated individual, the chances of spread or outbreak of the disease will be very less. This is so because if the contagious disease has to spread, the causative agent should infect new individuals, but When the majority of the people in the population are immunized, the infectious agents spread will be blocked. This way, the people who cannot receive vaccination, such as immunocompromised individuals or people in certain special clinical situations, are also protected. Passive immunization. If the vaccine contains preformed antibodies to protect the host as a preventive or curative measure against the pathogens, it is passive immunization. Behring and Kitasato were the pioneers in introducing passive immunization in 1890. They explained that immunity developed in one individual can be transferred to another individual by transferring serum. Administration of preformed antibodies provides immunity against the infectious agent in consideration. Preformed antibodies can provide natural as well as artificial passive immunity. The transfer of antibodies occurs naturally from mother to fetus through placenta, colostrum or mother's milk. Infant's immune system is not strong enough to fight against all types of infections. Antibodies produced by mother's immune system against diphtheria, tetanus, streptococci, rubella, mumps and poliovirus provides natural passive immunity to the baby. Passive immunization is done artificially also in the form of vaccines. This works as immediate remedy in certain situations. This is helpful in conditions where instantaneous treatment is required, such as in case of dog bite, snake bite, bee sting or accidental injury. Dog bite may introduce rabies virus, snake bite may spread poison in the whole body, bee sting can lead to fatal hypersensitive reactions and accidental injury can cause tetanus. In such cases, instant administration of antibodies can restrict the establishment of infection and damage to the body. These antibodies are raised in another animal such as horse. Passive immunization is also useful in case of people traveling to regions prone to certain infections and for healthcare workers. This method provides artificial passive immunity. Passive immunization provides only instantaneous relief and does not provide long-term immunity. Repeated immunization can lead to its lowered effectiveness and also certain hypersensitive reactions. As the antibody used for passive immunization is produced in some other individual, the recipient can mount immune response against it. Types of vaccines. Since the discovery of vaccination by Edward Jenner, attenuated and killed preparations of vaccines are used for prophylaxis. Understanding the detailed working of immune response has led to the development of new methods of vaccine production. However, the classical methods are still used. The vaccines can be categorized into classical and modern vaccines. The killed, attenuated and toxoid preparations are usually considered as classical methods of vaccine preparation, and recombinant, subunit, conjugate and DNA vaccines are considered as modern vaccine preparations. First, killed or inactivated vaccines. These are the whole cell vaccines in which the pathogenic microbe is killed by either heat or chemical treatment, so that it cannot cause disease inside the host. Killing the microbe eliminates the pathogenicity, but the structure of the organism is intact. So, our immune system can recognize the foreign macromolecules on it as antigens and elicit immune response against it. The antigenic structures should not be altered during inactivation process. Heat treatment may disturb the protein antigenic structures, which renders the microbial preparation useless. Treatment with formaldehyde or alkylating agents is preferred for inactivating microbial cells. They can also be irradiated by gamma rays. The inactivated Salk polio vaccine is prepared by treatment with formaldehyde. As the microbes are killed in these vaccines and cannot multiply in the host, repeated doses are introduced inside the host to increase the antibody titer, known as booster doses. Humoral immune response dominates in this case with lesser degree of cell mediated immunity. As compared to the attenuated vaccines, the risk of pathogens reverting back to virulent forms is absent. They are safer and stable because the pathogen is dead. They do not need to be refrigerated. Some inactivated vaccines used in routine are whole cell pertussis vaccine, hepatitis vaccine and inactivated polio vaccine. Second, live attenuated vaccines, LAV. These vaccines contain live pathogens which cannot cause disease because they are attenuated. Attenuation is a process in which virulence power of the pathogen is significantly reduced so that it is unable to cause disease. These are weakened cultures of pathogens. This was first observed by Pasteur when he was experimenting with causative agents of chicken cholera. He developed attenuated strains for treating anthrax. Attenuated cultures, when introduced inside the host, cannot cause disease, but can generate the immune response because of their intact structure. Our immune system recognizes the antigens on pathogen and develops immune response. The attenuated strain invades our body just like a true pathogen and primes the body for humoral as well as cell mediated immunity. As the organism is live, it multiplies inside the host, which leads to continuous increase in the supply of antigens to our immune system, and the antibody titer goes on increasing. LAV gives long lasting immunity. The immune response developed is excellent and comparable to infection with a wild type pathogen. In case of viruses and intracellular pathogens, prominent cell mediated immune response is developed. There are some disadvantages also associated with live attenuated vaccines. There is a possibility of attenuated strain reverting back to the virulent strain, although this risk is rare. The storage and transport of LAV requires refrigerated conditions, which may be difficult in developing countries. As the advantages of these vaccines supersede the disadvantages, numerous such vaccines are recommended by WHO. For example, tuberculosis BCG vaccine, oral polio vaccine, rotavirus, measles and yellow fever vaccine. These vaccines can be produced by attenuation of pathogens by various methods, such as growing the pathogen in non-susceptible host. Pathogenic viruses or bacteria, when grown repeatedly in non-susceptible host, adapts for replication in this host. Such a pathogen, when again introduced inside a susceptible host, exhibits lesser replication ability, which is insufficient to cause infection. Oral polio vaccine developed by Sabin was cultured in monkey kidney cells for its attenuation. Vaccination for tuberculosis is done by attenuating Mycobacterium bovis, which is closely related to the original pathogen Mycobacterium tuberculosis. The developed attenuated strain is known as Bacillus calmette-guerin (BCG). The yellow fever virus was attenuated by growing it in duck embryos for numerous generations to be used as a vaccine for yellow fever. Next method for attenuation is growing the pathogen on artificial medium for a number of generations. When a microbial cell is grown on artificial medium for a long time and subcultured several times, it tends to lose its properties. The characteristics which it exhibits in its natural habitat, such as soil, animals or human body, are in accordance with the environment. While growing on artificial medium, it does not need to produce virulence factors, which are required for invading a susceptible host. Therefore, it may lose the capacity to produce them. Pasteur was the first one to notice this. While experimenting with chicken cholera, he accidentally inoculated old cultures in his experimental birds and found that they did not catch the infection. When he again tried to develop infection in these previously inoculated birds with fresh cultures, they were not affected. He concluded after repeated trials that old cultures have immunized the birds and the original pathogenic strain are now not able to cause infection. Growing the pathogen in presence of an inhibitory substance. Certain inhibitors are known to suppress the production of virulence factors of a pathogen. Hydrolyzing enzymes are important virulence factors of pathogens which can be inhibited. Attenuated strains of Pseudomonas aeruginosa have been developed by inhibiting its quorum sensing. Next method for attenuation is growing the pathogen at elevated or suboptimum temperatures. Pathogens' virulence and growth is at its best at optimum temperature. They lose their characteristics by growing them at elevated or suboptimum temperatures. Next method, desiccation of the tissue containing pathogen. Prolonged desiccation of the tissue containing pathogen leads to its neutralization. This preparation was used by Pasteur to prepare a vaccine against rabies. He transmitted intracerebral inoculations from one animal to another and then desiccated the spinal cord of the infected animal until the preparation became almost non-virulent. The preparation was completely neutralized, which resembled inactivated vaccine. Next, genetic engineering of the pathogen's genome. Pathogens can be attenuated irreversibly by removing the virulence factor genes by genetic engineering. Herpes virus vaccine for pigs has been prepared by removing thymidine kinase gene responsible for its virulence. Third, toxoids. Some pathogens cause disease inside the host's body by producing toxins. In such cases, immunity can be provided by exposing immunogenic structures of toxins without introducing pathogen inside the host's body. The pathogen does not cause harm directly, but destroys the host tissues or organs by producing exotoxins, such as in case of tetanus and diphtheria. These exotoxins are highly immunogenic and harmful for the host. Toxins can be inactivated by treatment with formaldehyde and converted into toxoids. Toxoids are also immunogenic in nature and can generate the immune response, but cannot cause harm to the host. Toxoids are administered as vaccines inside the host's body. The antibodies produced in response to the toxoids can also neutralize the toxins and are known as antitoxins. These are effective in destroying the exotoxins produced by pathogens in later infections. The exotoxins can be produced without the interference of a pathogen by genetic engineering. The gene responsible for the production of exotoxin can be cloned and expressed in a host. This can be eventually purified and inactivated to form toxoids. Toxoid vaccines are routinely used for prevention against diphtheria and tetanus. Conclusion. Since the advent of vaccines, numerous infectious diseases have been controlled to a great extent. the rate of occurrence of outbreaks and epidemics has reduced drastically. Following the prescribed vaccination schedule may lead to eradication of many more diseases like smallpox and polio. Advancements in the field of vaccinology may also soon give us some solutions to dreadful diseases like AIDS and cancer.